I guess if anybody else comes in on this side, you can just pass that around to them. Okay, it looks like it's about time to get started. First of all, let me stop the share for a second. Let me just do a quick audio check to make sure everybody can see and hear me. So, okay, I'm getting a thumbs up. Good. I can mute this again. Okay, so today is your review session for exam number 2. Which is Monday morning during our usual class time at 9. So again, just to remind you, this is going to be given online, so you should not show up for class on Monday, but rather make sure that you have access to the internet, for the usual class time. I'll do it the same way I did last time, so you'll have a window of about 15 minutes or so. 20 minutes. I'll let you start at about 8:55, and you'll have until 9:15 to begin the exam. After you begin, you'll have 30 minutes to complete the exam part. Okay, the exam part is going to be 40 questions, multiple choice, worth 2.5 points apiece. And then separately, there will be a bonus question worth 5 points. Okay, and you'll have 8 minutes to complete that. I think that's about it. So for the rest of the period, I'll be happy to answer questions on anything you want me to go over again, anything that you have questions on. Okay, so I guess with that, I'll just open up the floor for any questions. Anybody in the classroom or on Zoom have a question about anything we covered since the last exam? Yes? Sure. Endergonic versus exergonic reactions. So that's from chapter 5. Let me just pull up that PowerPoint. Okay, so looking at exergonic versus endergonic reactions. Exergonic reactions are reactions where there's less free energy in the products than the reactants. Okay, so if we look at, so here's an example of an exergonic reaction. So the reactants up here have high free energy, and the products down here have low free energy. Okay, again, free energy is simply the amount of energy in the reactants or the products that's available to do work. Okay, so in this case, we have high free energy in the reactants, we have low free energy in the products. Okay? So when we go from high energy reactants to low energy products, we get energy out. Okay, and again, you can think of this as a ball rolling downhill. So we put a ball on top of a hill that has potential energy. When the ball's at the bottom of the hill, it has low potential energy, high potential energy when it's at the top of the hill. So we're getting energy out in the form of kinetic energy when the ball rolls downhill. Same thing, so going from high energy reactants to low energy products is like the ball rolling downhill. We're getting energy out in the form of, well, it depends, in the form of heat or energy to do work. Okay. And again, let me just reintroduce this term to you. So G is how we abbreviate free energy. So we have G on the y-axis. And this change in free energy from high energy reactants to low energy products, we refer to as the delta G. Okay, because we're going from high energy reactants to low energy products, we have a negative delta G. Okay, and negative delta G means exergonic reactions. Anytime we see a negative delta G, we know that that's an exergonic reaction, and that reaction will tend to happen spontaneously. Okay. On the other side of the coin, if we look at endergonic reactions, in endergonic reactions we have the opposite. We have high energy products here, and we have low energy reactants down here. So in order to drive this reaction forward, we need to put energy in. Okay, this could come from a couple different sources. We could hydrolyze ATP. ATP is energy currency of the cell. So we could hydrolyze the high energy bonds from ATP for energy to drive endergonic reactions forward. Photosynthesis, we could use the energy from sunlight to drive endergonic reactions forward, right? So again, endergonic reactions, we're going from products, we're going from reactants with low free energy to products with high free energy. I'm making a mess of this here. Okay, so in this case, the change in free energy, or delta G, is going to be a positive value. Whenever we have a positive delta G, that means that the reaction is endergonic and will tend not to happen spontaneously. So again, we can think of this, if we use the ball analogy again, a ball sitting down here has low potential energy. A ball sitting up on the top of the hill is going to have high potential energy, so high potential energy up here. In order to get it from down here to the top of the hill, we need to put energy in. We need to push that ball uphill. So that's endergonic, energy in order to drive that reaction forward. Okay, I think, did that answer your question, or should I go further? Okay, good. All right. Other questions? Anybody from Zoom have anything? Can you go over the light and dark reactions of photosynthesis? Sure. Okay, so why don't we start here with the overall reaction. So the overall reaction of photosynthesis is taking 12 H2O plus 6 CO2 plus the energy from light and making glucose, oxygen, and 6 H2O, okay? So again, this is a biosynthetic reaction. This is remember, we can divide metabolic pathways into two types: anabolic pathways and catabolic pathways. Okay, this is an anabolic pathway. So this is where we're taking small, low-energy reactants, in this case, water and CO2, and we're making large, high-energy products, in this case, glucose, C6H12O6. Okay, so we're building, we're making something bigger. So an anabolic pathway will tend to have endergonic reactions. Okay, we need to put energy in to build these bigger, higher energy molecules like glucose. Okay, so we can divide photosynthesis into two parts. We have the light-dependent reactions and the light-independent reactions. Okay, in the light-dependent reactions, we're going to make some important stuff that we're going to need in the light-independent reactions. Okay, so we're going to make ATP in the light-dependent reactions. That's going to provide energy for making glucose from CO2, and it's going to make, we're going to get NADPH. Okay, NADPH is a reduced cofactor that's going to provide electrons and hydrogens for making glucose in the light-independent reactions. All right, so as if you guys aren't sick of hearing me go through this, let's go through this one more time. What happens in the light-dependent reactions? So the enzymes that do photosynthesis are embedded in the innermost membrane of the chloroplast. Okay, does anybody recall what that's called? The innermost membrane. So it's folded into these stacked discs. Nobody recalls that? The innermost membrane is called the thylakoid membrane. And you might recall that the thylakoid membrane is stacked. Well, let me show you. There. Okay, so here is the chloroplast. The chloroplast has three membranes, two outer membranes, and a very highly convoluted, highly folded inner membrane called the thylakoid membrane. Photosynthetic pigments, like chlorophyll, and the electron transport chains of photosystem 1 and 2, all of these, the reaction centers, all of these are embedded in the membranes of the thylakoid membrane. So all of these are in the thylakoid membrane. All right. So what we're looking at on this slide, okay, this is a cross section of the thylakoid membrane, and we can see all of these. We can see the photosynthetic pigments and the electron transport chains. Okay. So we can divide these up into two photosystems. We've got photosystem 2, which consists of a light harvesting complex of chlorophyll molecules. Inside this light harvesting complex, there is a reaction center. The reaction center of photosystem 2 is called P680. Oh my God, it's so hard to draw with this. There we go. Okay, so we have light harvesting complex, reaction center, P680 in this case, and we have an electron transport chain. Photosystem 1, again, we have a light harvesting complex. We have a reaction center. In this case, the reaction center is called P700. Right. And then we have a second electron transport chain. Okay. And also very important not to forget, we have this enzyme called ATP synthase over here. All right, so going through the light-dependent reactions again. The light-dependent reactions start when the light harvesting complex of photosystem 2 captures a photon of light. Okay? A photon of light is simply a particle of light. Again, light's one of those weird things in physics. Sometimes it acts like a wave, sometimes it acts like a particle. In this case, it's going to act like a particle. Okay? So that particle or photon of light is going to bounce randomly between the chlorophyll molecules in the light harvesting complex until it encounters the reaction center, which is P680. Once it encounters the reaction center, that's going to put 2 electrons in a higher energy state, and those electrons are actually going to be ejected from P680. Now we need to replace those 2 lost electrons. For photosystem 2, the way we replace those 2 lost electrons is to split a molecule of water into 2 electrons, which replace the 2 lost ones, 2 protons, and 1/2 of an O2 molecule. Okay, now the protons, of course, carry a charge, meaning that they can't cross the phospholipid bilayer. So they're going to accumulate in here, and they're going to start to build up this concentration gradient that we can use to drive ATP synthesis. The 1/2 of an oxygen is going to join with another 1/2 of an oxygen. Okay, that's small, nonpolar, uncharged, so that can diffuse across the plasma membrane and go away here. Going back to the electrons, so the 2 electrons lost from P680 when they entered a higher energy state are going to enter an electron transport chain. So as these electrons are passed from component to component in the electron transport chain, they're going to go to lower and lower energy states. And the cell is going to capture some of that energy from electrons going to lower energy states and use it to do work. Specifically, the work that's going to be done here is it's going to power a proton pump. So the energy from electrons going down their concentration gradient is going to be used to pump a proton from the stroma. Okay, this is the stroma. The stroma is the space rather, between the thylakoid membrane and the outer membranes into the thylakoid compartment. Here, okay, the thylakoid compartment, this is the inside of the thylakoid membrane. So again, we're building up this proton concentration gradient that we can use to drive ATP synthesis later. That brings us to photosystem 1 rather. So photosystem 1, pardon me, same thing happens. So the light harvesting complex captures a photon of light, which bounces randomly between all of the chlorophyll molecules in the light harvesting complex until it encounters the reaction center, which is P700 in this case. Again, 2 electrons are going to enter a higher energy state and be ejected from the reaction center. Again, we need replacement electrons. This time, the electrons from the electron transport chain of photosystem 2 replace the 2 lost electrons from P700. All right. Now, those 2 excited electrons are going to enter a shorter electron transport chain over here. At the end of this shorter electron transport chain, again, we're going to use that to do work. This time, the work that's going to be done is to reduce a molecule of NADP to NADPH. Okay, so that's right here. So a proton plus NADP plus electrons from the electron transport chain make NADPH. Again, this is going to come in handy in the light-independent reactions where we use these electrons to make glucose from CO2. Finally, the whole time we've been doing this, we've been building up this high concentration of protons in the thylakoid space. Okay. So the enzyme ATP synthase, which is over here, okay, this has a channel that the protons can go down. They can exit they can exit the thylakoid compartment through this channel in ATP synthase. Okay? So of course, protons are going to want to go down their concentration gradient out of the thylakoid compartment. As they go down their concentration gradient, some of the energy is captured to drive ATP synthesis. ATP synthesis. So that drives this endergonic reaction, right? So we're taking ADP plus inorganic phosphate plus the energy from protons going down their concentration gradient, and we use that to make ATP, which of course is the energy currency of the cell. Also, this is going to be important in the light-independent reactions because we're going to need that energy to drive the endergonic reactions involved in making glucose from CO2 and water. All right, so that is the light-dependent reactions. Let's look at the light-independent reactions next. Let me just clean this up a little bit, I'm making a mess. Okay, so in the light-dependent reactions, this is where we're actually going to make we're going to make, we're going to make glucose from CO2. So ATP provides energy for driving these endocytic reactions forward. So we use ATP from the light-dependent reactions. NADPH acts as a donor of electrons and protons. So we're going to use the NADPH that we made in the light-dependent reactions. And also, we need CO2. If CO2 from the air is going to provide a source of carbon and oxygen for making glucose. We can divide up the light-independent reactions, we can divide it up into 3 stages. So we have the carbon dioxide fixation stage, or the carbon fixation stage. We have reduction of PGA to PGAL. And then remember, this is a cycle. Another word for the light-independent reactions is the Calvin-Benson cycle. So just like the Krebs cycle, we have to regenerate one of the molecules that we started with so it can go back into the cycle. So the last stage is regeneration of RUBP, that's ribulose bisphosphate, from PGAL. Okay, I'm using the abbreviations from the next slide. I like this figure, but I don't like the terminology. So G3P, that should be PGAL, and 3-phosphoglycerate, we abbreviate this PGA. Okay, so going through these 3 stages, so first we have to fix the carbon from carbon dioxide. So in the first stage, we join together a carbon dioxide with this 5-carbon molecule called RUBP, or ribulose bisphosphate. That makes a 6-carbon molecule, but a 6-carbon molecule that's extremely unstable. And almost immediately after it's formed, it breaks into 2 3-carbon molecules. Okay? 2 phosphoglycerates, 2 phosphoglycerate molecules, or PGAs. Okay, so that's the first step. That's the carbon fixation step. Next, we have to take PGA and turn it into PGAL. Okay, we do that by reducing it. So we're going to take energy from ATP, and we're going to take electrons from NADPH, and we're going to reduce PGA into PGAL, okay, PGAL. 2 things we can do with PGAL. So if PGAL is a 3-carbon molecule, so we can take 2 3-carbon PGALs, join them together, and make a phosphorylated glucose. Okay. Then from that phosphorylated glucose, we could use that to make more complex carbohydrates. So we could join together those glucoses to make starch, or we could join them together to make cellulose for plant walls, etc. Okay, so that's one thing we can do with the PGALs. Another thing that we have to do, though, is we have to remember this is a cycle, so we have to regenerate that 5-carbon ribulose bisphosphate. So some of this PGAL plus energy from ATP from light-dependent reactions is used to regenerate the 5-carbon ribulose bisphosphate, and this completes the cycle. And that 5-carbon ribulose bisphosphate goes back into the cycle, and this repeats. And as we're repeating this cycle, we're pumping out more glucose the whole time. Okay? All right. So Ian, did that answer your question? Did I miss anything? Yes. Okay, good. All right. Any other questions? Not seeing anything in chat. Anybody in class have anything? So let's go back and look at the study guide. So are you guys good with, so chapter 7 is a pretty difficult chapter as well. You have any questions on chapter 7? Glycolysis, PDHC, Krebs cycle, oxidative phosphorylation. You guys are good with all that? Anaerobic respiration? No questions on that?
Show us where each of the ATP comes from.
Oh, sure. Let me go back and do the board. Now, it's actually pretty fortunate that you asked that question because that's actually one of my favorite bonus questions is for you guys to go through the ATP accounting of anaerobic respiration. So let's go through that again. And let me just quickly open up a notepad document so I have on the board. Okay, good. I have a good background to draw on now. Okay, so again, I did this at the end of chapter 7. Now, what we're doing is a little bit of accounting. Okay, so we're going to account for all of the ATP that we make at each stage of aerobic respiration and each of the reduced cofactors, and then we'll add up all the cofactors, all the ATP that they can make in the electron transport chain. Okay, so first stage of aerobic respiration is glycolysis. Second stage is the Krebs cycle. And to get from glycolysis to the Krebs cycle, there's this intermediate step called the pyruvate dehydrogenase complex, or PDHC. So during glycolysis and the Krebs cycle, we're going to make some ATP by substrate level phosphorylation before we get to oxidative phosphorylation, before we get to the final step. We're going to reduce some NAD to NADH. And we're going to reduce some FAD to FADH2. So in glycolysis, let's just go over the steps of glycolysis really quickly. So in glycolysis, we start with a 6-carbon glucose molecule. We need to invest 2 ATP in order to get this metabolic pathway started. After we get the pathway started, we're going to make 4 ATP. So what's our net gain of ATP if we burn 2 to get this started and get 4 back? 2. So we get a net gain of 2 ATP. Also, during glycolysis, we're going to reduce 2 NAD to NADH. We don't reduce any FAD to FADH2. And we're going to end with 2 3-carbon molecules of pyruvate. Okay? That brings us to PDHC, pyruvate dehydrogenase complex. So in PDHC, we start with a 3-carbon pyruvate, and we end with a 2-carbon acetyl-CoA. So we're going to consume a molecule of coenzyme A, or CoA. We're going to reduce an NAD to NADH, and we're going to make a molecule of CO2. Okay? So since we get 2 molecules of pyruvate from glycolysis, this is going to be times 2, right? So times 2 means that we're going to per glucose molecule, we're going to get 2 NAD reduced to NADH2. We don't make any ATP, and we don't reduce any FAD to FADH2. Okay. So in the Krebs cycle, so remember in the Krebs cycle, we're going to start with a 2-carbon acetyl-CoA and a 4-carbon oxaloacetate. All right? For each time we go through the Krebs cycle, we're going to reduce 3 NAD to NADH2. Scratch that. We're going to reduce 3 NAD to NADH, and we're going to reduce 1 FAD to FADH2, and we're going to make 1 ATP by substrate level phosphorylation. Okay? But remember, we got 2 acetyl-CoA from the pyruvate dehydrogenase complex. Okay, so again, everything is times 2. So that means we're going to make 2 ATP by substrate level phosphorylation. We're going to reduce 6 NAD to NADH2, and we're going to reduce 2 FAD to FADH2. All right, so adding all this up, that's 4 ATP by substrate level phosphorylation. Now in the electron transport chain, we have 10 NAD, we have 2 FADH2. Each time an NADH gives up its electrons to the electron transport chain, we're going to make 3 ATP per NADH2, right? So that means that these 10 NADH are worth 30 ATP in the electron transport chain. Okay? Each time an FADH2 gives up its electrons to the electron transport chain, we're going to make 2 ATP. So these 2 FADH2 are worth 4 ATP in the electron transport chain. Okay, so adding up the ATP we make by oxidative phosphorylation here and the ATP that we make by substrate level phosphorylation, we get 38 ATP. ATP total. And again, you guys might remember, pretty much all semester I've been telling you that when we metabolize a molecule of glucose by aerobic respiration, we get a net gain of 36 ATP. Right. Where do the 2 extra ATP go? Well, remember that glycolysis takes place in the cytoplasm. PDHC in the Krebs cycle and the electron transport chain as well all take place in the mitochondria. Okay, so we have to get these 2 NAD that we reduced from glycolysis from the cytoplasm into the mitochondria. Okay, and we need to do active transport to do that. So we have to burn 2 ATP to transport these 2 NAD into the mitochondria. However, I mean, in the electron transport chain, these 2 NAD are worth 6 ATP, right? So we're still getting a net gain of 4, even though we have to transport them into the mitochondria. Okay, so that's where the 2 extra ATP go. Okay, did that answer your question? Good. Okay, and again, this is one of my favorite bonus questions, so you guys probably are going to want to study up on that. Okay, 9:33. There's time for probably 2, maybe 3 more questions, depending on how long they are. I'm not seeing any questions from the classroom. Anybody on Zoom have any questions? Okay. Well, I got a private message. I don't think, since it's a private message, I probably shouldn't read that out loud. I'll get back to the person who gave me a private message. But on that note, do remember to study your quizzes. Remember that I do take at least one question from each quiz and put it right on the test. So that's a good study guide for you, a good study resource are your old quizzes. All right. Question, if we only have 45 seconds to answer each question, does that mean we won't be able to double check our work at the end? It should be fine. This is from Stephanie. It should be fine. 45 seconds is a lot more time than you think. As long as you're familiar with if you've studied and you're familiar with the material, there shouldn't be any problem. Most students finish these tests in 20 to 30 minutes anyway, so it really shouldn't be a problem. Hannah has a question. Could you go through oil rig and Leo the Lion Goes Grr abbreviations? Sure. Let's go back to the whiteboard again. Keyboard again. Okay, so Leo the Lion Goes Grr and oil rig, these are what are called mnemonic devices. Mnemonic devices are little phrases or abbreviations that help you to remember things. What these two mnemonic devices help you to remember are oxidation and reduction. Okay, so the abbreviation oil rig stands for oxidation is losing electrons and reduction is gaining electrons. Okay? Leo the Lion Goes Grr. So Leo the Lion Goes Grr. This is just a different mnemonic device for the same thing. Okay, Leo stands for losing electrons is oxidation. Grr stands for gaining electrons is reduction. Okay, let's look at some examples. So I've been talking about cofactors an awful lot. So cofactors are molecules that help enzymes, often by carrying electrons. Okay, so carrying electrons, that's going to mean that they're going to be reduced, or by carrying functional groups. So we've talked about the cofactors NAD, FAD, and NADP. Okay? So this is what they look like when they're not carrying electrons. When they're helping an enzyme by carrying electrons, they also, it's easy to tell if they're carrying electrons because the electrons come along with hydrogens. Okay, so if we add 2 electrons and a proton to NAD, we get NADH. If we add 2 electrons and 2 protons to FAD, we get FADH2. And if we add 2 electrons and a hydrogen, or a proton rather, to NADP, we get NADPH. Okay, so again, let's look at these mnemonics. So oil rig, oxidation is losing electrons, reduction is gaining electrons. Okay, so when NAD gains electrons, it's being reduced. Okay, so this is the reduced form of these coenzymes. Now, if we were to do the opposite thing, if we were to give up these electrons, say to the electron transport chain in oxidative phosphorylation, okay, we would be oxidizing NADH back to NAD, okay, because we're losing electrons. Okay, losing electrons is oxidation, okay? So reduction, gaining electrons, this direction is reduction. If we do the reverse reaction and we lose electrons, that's oxidation. Did that answer your question, Hannah? Okay, yes. All right, so yes.
Could you go over carbon fixation?
Carbon fixation, okay. Let's, so that's photosynthesis. There we go. Okay, so carbon fixation is simply this first step in the light-independent reactions. Okay, so this is the carbon fixation step, where we take the 5-carbon ribulose bisphosphate plus the 1-carbon CO2, and that makes the 6-carbon molecule that breaks into 2 PGAs here. Okay, so that's simply what carbon fixation is. Now, it's a little bit different. Remember that we have 3 different light-independent pathways. Okay, whoops, I want to word this. Okay, so we have 3 different light-independent pathways, and carbon fixation is a little bit different in the 3. So the Calvin-Benson cycle, the carbon fixation route that I showed you, is called the C3 pathway, because we fix the carbon as a 3-carbon PGA. We also have the C4 pathway. So in the C4 pathway, carbon fixation is a little bit different. Okay, it's fixed as a 4-carbon molecule. We're not going to go into all of the reactions. You guys don't have to know all the reactions in the C4 pathway, but just know that carbon fixation is a little bit different in the C4 pathway. So we have carbon fixed as a 4-carbon molecule. And then again, it's actually very different in the CAM pathway. The CAM pathway uses a very different method of carbon fixation. And again, you guys don't need to know that pathway. You just need to know that the CAM pathway is very different, and we can fix carbon and store it for long periods of time, which means that these CAM plants are very, they can store lots of water, and by not opening up their stomata during hot, dry parts of the day. Okay, looking at the time, I think we have time for one more quick question if you have one. If not, yes?
Can you explain the difference between mutase and kinase?
Mutase and a kinase, sure. Let's look at the whiteboard again. Okay, so the question is, what's the difference between a mutase and a kinase? A kinase adds a phosphate. So the definition, I think, on the slide is it catalyzes a transfer of a phosphate group from one molecule to another. Basically, what it does is it adds a phosphate. A mutase rearranges a phosphate within the same molecule. So it just takes a phosphate from one part of the molecule, moves it to another part of the molecule. That's what a mutase does. For a kinase, a molecule that doesn't have any phosphates, a kinase adds a phosphate to that molecule. That make sense? Okay, and it looks like we're out of time. So you folks have a nice weekend. Be careful. It seems like in my email, I'm getting more and more notices about COVID-related events. So guys, don't let up. Be vigilant. Wash your hands. Wear your mask. Do social distancing. Have a good weekend, and good luck on your exam Monday morning.
other departments from the